Well, let's go ahead and get started. Welcome, everybody. I'm going to introduce today's seminar speaker, Dr. Craig Galban. Craig Galban is professor of radiology and biomedical engineering here at Michigan. He did his PhD at Florida State and postdoctoral work at the NIH, and he started as an assistant professor here in 2008. He has more than 100 publications and has won numerous awards, including being named a distinguished investigator of the Academy of Radiology Research. His research focuses on image feature extraction for pulmonary disease phenotyping, which I believe he'll talk about some today. And he also recently affiliated with DCMB. So Craig, thanks for talking to us today, and we're excited to hear about your work.
All right, thank you. Thanks for the kind introduction. So yes, today I'd like to thank you all, for those of you who came in person. Fists up, well done. For those online, I really appreciate you chiming in or being on board. And this is a kind of a project we started relatively recently, investigating the use of clinical trajectory analysis for analyzing a lot of the image-based techniques that we develop and determining if they are risk factors of COPD. Yeah, there you go. Okay, wrong way. All right, yes, here's my disclosure. Somebody help me out here, I'm panicking, drowning. So yes, some of the technology that I'll be presenting in this talk is licensed to a medical software company called InBio. So I promise not to be too ecstatic. So it's up to you guys to determine which software it is. So instead of giving, normally I give an outline of how the talk is going to play out, but I decided just give an overview. Something that we've been working on for quite some time is developing novel image-based readouts for improved diagnostic and prognostication in COPD. It's really through a recent collaboration that was brought to my attention through one of my postdocs that's really kind of opened the door and how this data can be analyzed, and we can fully exploit the breadth of data that's available through what's called the COPDGene, and I'll describe in detail. So this is that study, and again, it's sort of in its infancy, but we feel like we're making strides. So COPD, why COPD? What is COPD? COPD means chronic obstructive pulmonary disease, and it continues to be the fourth leading cause of death in the United States. About 50 million people are diagnosed with COPD. About 12 million are walking around today, not even knowing they have it. So this is a huge health and financial burden to the US. As many of you can kind of guess, smoking continues to be the main culprit and why COPD is a problem. But it's not the only culprit. Environmental factors, occupational hazards, as well as genetic factors, all can contribute to the development of COPD in patients.
Can we ask a quick question?
Oh, no, absolutely, please.
I just wonder, is vaping associated with it as well?
That's a great, it's so new. A lot of that research hasn't really come through. So COPD is sort of a long process, and there are pulmonary diseases associated, damage, but they may not necessarily be COPD. So I'm not familiar. But I think we have room for an R1. I think we should talk later. So what is chronic obstructive pulmonary disease and how is it assessed? Meaning, how do doctors diagnose it? Well, COPD is it's sort of an umbrella name for a variety of different subsets of diseases. And the primary cause is the inhalation of some sort of insult that causes an inflammatory reaction in the lungs. In the case of the two main that I'm referring to now is bronchitis and emphysema. In the case of bronchitis, the airway walls become inflamed. They start producing a mucus, essentially to remove this inhalant. And but because it's continuous, say like smoking, it becomes chronic. The mucus begins to build up, the airways begin to swell, the luminary begins to tighten, restricting the flow of air out of the lungs. This is reversible, meaning if you stop whatever you're doing or get out of whatever you're breathing in, this process should correct itself over time. Unlike bronchitis, emphysema is a one-way street. So once it starts, it doesn't stop. What emphysema is again, you're exposing the tissue to something, you're eliciting an immunological response. But what happens is the immune system begins to start breaking down the alveoli. And I hope everyone can see my mouse. And this is what the airways, the alveoli should look like. They look like little grapes. This is where all the gas exchange is happening. And ultimately what happens as the immune cell is just activated, it begins tearing down these walls, and you begin to get what's called alveolar enlargement or airspace enlargement. But not only is the alveolar beginning to break down, the interstitium is also beginning to break down. So you're losing a lot of the elasticity. So essentially, you're going from a balloon that you can inflate and it deflates through its elasticity. You essentially just have a plastic bag. Ultimately, in both processes, you can inhale, but you can't exhale. Your lungs are essentially filled with air. So how do I'm not a medical doctor, don't get sick. How do physicians manage these patients, diagnose? Well, these patients are going to come in, they're going to be coughing, wheezing, they're going to feel fatigue, they're going to feel chest pain. The first thing that's going to happen is they're going to be evaluated, their medical history. Does your family have a predisposition? Are you a smoker? That's the obvious. Do you work in a mine? These are the questions that are going to be asked. The next is, if it's severe enough, you may undergo what's called a pulmonary function test or spirometry to determine how healthy your lungs are. How much air can you move out of your lungs? And if things look worse, and it really, but this is like a global measure, but if that's poor, you may undergo what's called an X-ray computed tomography. Now, an X-ray computed tomography is the three-dimensional version of an X-ray you'll get if you break a bone or you're getting your teeth examined. It's essentially a density map, a three-dimensional density map. And just to kind of help people guide the eye, these are the lungs, and the airways are black because they're very low density. The soft tissue is gray, and the bones are white. So we're looking at density. And radiographically, a trained radiologist could diagnose and identify bronchitis and emphysema fairly rapidly. But there are also quantitative indices in readouts that can be generated from these CT data set. Emphysema can easily be identified and quantified using CT and not even super advanced techniques. But bronchitis also, we have techniques that can remove the airways from these images, and we can measure the airway wall, the lumen area, and the wall area percent, which is a readout of bronchitis. What has been missing for quite a while is the ability to measure what's called bronchiolitis. And like its big brother, bronchitis, it is the inflammation of the terminal and respiratory airways. So instead of the larger airways, they're the airways right before the alveoli. And about 90% of all airway limitation in mild to moderate COPD is contributed to that airway, that dysfunction or that subtype. But because these airways are so small, they're invisible on computed tomography. They're invisible on CT. You just can't measure it. So the NIH, and more importantly, the NHLBI, the National Heart, Lung, and Blood Institute, understand the significance of tackling COPD. It is a national problem and a global problem at that. So like all things government, they poured a lot of money into a worthwhile initiative, and I think it is worthwhile. We're getting a lot of info out of this into large multi-center trials. And these multi-center trials are not simply just to collect the data, but to provide clean, sort of reproducible cohort of data, a data repository where essentially can be outsourced. So minds across the United States as well as the world can access this to kind of combat and identify, if there are unique subtypes, how would you treat those subtypes? How would you, if you can improve diagnosis of the disease and you can improve management. So COPDGene is one of these large multi-center trials with the main goal of this trial was to characterize and identify genetic as well as CT-based subtypes for COPD, as well as patients at risk of developing COPD. So we're talking about heavy smokers. And this is quite a big trial. There are over 21 sites across the United States, or I believe it is 21 sites across the United States, not over. Phase I that started back in 2008 accrued over 10,000 subjects, and this is now complete. Phase II, about half of the 10,000 came back for a 5-year follow-up, and that's also completed. That recently completed, and now we're currently into phase III. So far, and last I checked, we're at about 2,500 subjects. We may be at more. So let's say about a quarter of the initial 10,000 came back for a 10-year follow-up. So this data is not just a large amount of a large number of patients that were accrued or cases that were accrued, but we had data like demographic, spirometric, respiratory questionnaires, as well as computed tomography and genetics available in a metadata. So what was the Michigan Medicine's contribution? I was fortunate enough to be introduced to one of my main colleagues here involving COPD, Meilon Han, and we developed a technique that allowed us to quantify and visualize bronchiolitis, that invisible component of or subset of disease in COPD. And we termed this methodology called Parametric Response Mapping. And it is a purely computational technique that's applied to CT data. And just to kind of, I'm not going to go in gory detail, but it's important to understand what the technique does, is we take inspiratory CT data, biphasic CT data, so CT data acquired at full inflation and CT data acquired at expiration, so deflation. And both of those data sets provide some sort of information on the pathophysiology of the disease, such as emphysema and bronchiolitis. So, but because the two images are at different volumes, we spatially align the data to remove the spatial dependence. So now each individual dot, like blue dot here, is a point in space. And it consists of an expiratory CT value and an inspiration. HU is just the unit of measure for CT. Think of it as density. So you have two densities for each point in space. Then we apply a classification model to classify each of the individual voxels as normal lung, emphysema, small airways disease, which in this case is bronchiolitis, and parenchymal disease, which is sort of a catch-all for fibrosis, interstitial lung diseases, pneumonitis, stuff like that, which you wouldn't necessarily expect in COPD. And this is what it looks like once you've classified it. And because this data is spatially aligned, you can visualize it spatially. So we could see where small airways disease is occurring in the lung. But for the context of this talk and this work, we're simply going to take all the voxels that are classified as small airways disease, sum them up, and normalize it to the total lung volume to get a percentage. So say 30% small airways disease, 20% emphysema. We're going to treat these values as scalar quantities and for the context of this. But just to kind of give an idea of the breadth of the data that we're extracting, this is a gold forceps. So somebody with severe COPD, the yellow is small airways disease and the red is emphysema. So this is pretty bad. Okay.
So that's fairly acute towards the end of life.
Yeah, gold 4 is pretty bad. You're on a ventilator at that point. So what are the major finds of this work? This paper came out in 2012 in the Journal of Nature Medicine, and we found that small airways disease, as measured by this technique, is an independent readout of clinically meaningful measures, and also a precursor of emphysema. So as far as our results of small airways being independent, we did a bivariate regression looking at clinically relevant measures. And we found that in that bivariate model, when we had small airways with emphysema, it wasn't simply that small airways was a mild emphysema or something like that, but it was it contributed to the statistical significance to the correlation to the individual readout. So it was seen as an independent readout. So that was important. But what was really exciting, or at least we still think it's kind of exciting, is just to kind of guide the eye. The x-axis is the percent volume of emphysema, so 20, 30%, for example. And then on the y-axis is the percent volume of FSAD, 20 to 30%, again, of the total lung volume. If you look at the data, and the data is color coded, this is all still COPDGene. These are only a COPDGene trial, and we color code each individual point as a case, and we color code it based on disease severity. NA is patients at risk, gold 4 is the most severe. Right away, I'm going to guide your eye. I probably don't have to, but you can see that there's a trend in the data. For those patients at risk and with mild COPD, pretty much dominated by small airways disease. As the severity increases, you begin to see more emphysema, and then eventually the small airways disease plateaus and begins to drop and just dominates. Emphysema begins to dominate and be the what's considered a dominant emphysema-dominated COPD case. So if we looked more closely, based on this data, we suggested that, well, possibly small airways disease is a precursor to emphysema, that if we know how much small airways somebody disease has, we may be able to predict the onset of that irreversible process, which is emphysema. Well, we did a little bit more digging in the literature before we published, and we found that someone smarter than myself, Jim Hogg out of Vancouver, came up with the same conclusion, that using micro CT pathological samples, core samples of lung tissue, he found that small airways disease actually is a precursor, an intermediate to emphysema. So at the very least, we corroborated their results. So just to, the next big find, kind of jumping ahead, is we found that for those patients, this is another COPDGene study. Here we were interested in determining, does the extent of small airways disease predict spirometric decline? Meaning, if you have a lot of small airways disease, will your lung function decline over a 5-year period? And in fact, that's exactly what we see. And it's only in those cases at risk of developing COPD did we see the strongest effect. So just to recap, healthy lung, you do not want to follow this path. You do not want to go from healthy lung to emphysema. Emphysema, the only cure or treatment is surgical resection or lobectomy. You have to just remove that. Emphysema is irreversible and is a major cause of morbidity and mortality in COPD patients. Jim Hogg, and we corroborated his work, found that small airways disease, which is an irreversible process, it may serve as an intermediate to a worsening condition. In that case, emphysema. Parametric response mapping gives us the ability to visualize and quantify all 3 phases of the lung with regard to this simple model. So what we were interested in doing is, using PRM, which gives us the opportunity to really evaluate all 3 components simultaneously. So that's what we have in front of us. That's where we've gotten. But now we really want to use the full breadth of the COPDGene data set in as close to an unsupervised approach as possible to better understand the role of PRM-defined FSAD as a precursor to COPD. And that's what this discussion will be on. So before I start going in there, still have a little bit more background, is that the model that we're using, the clinical trajectory analysis technique, is based on the principal component, more specifically, principal graphs and manifolds. This is principal component was invented by Carl Pearson back in 1901. The idea is that you can have these components to represent the majority of your variants without having to use all the data. It's our collaborators, Alexander Galban and Andrei Zinovsky, really did a lot of the legwork in terms of developing techniques and elastic principal graphs and manifolds. To kind of give an idea of what that is, the idea is that this multi-dimensional data set is a nonlinear function. But if you sort of zoom in very close, you can define your three-dimensional space using a multitude of linear-based PCs. And here's an example of what that would look like, this manifold, and you can see it in a two-dimensional space. But what we're interested in is using what's called branching principal components. Here, the idea is we can have a simplified, very complex data set, and we can represent it using this approach, but by having very specific points, in this case, the yellow dots as nodes, and I'll describe this in more detail. Here's just an example of iris, the flower phenotypes, and how they're related among one another through this branching. Okay. I'm trying to get the movie to work. There you go. Awesome. Oh, it's getting dry. So this is some of the work on branching principal components. Again, the idea here is you can have a complex data set, nonlinear, and you can represent it through these through this iterative process of identifying more and more local PCAs that best represent your data. So this is the strategy we're using. And back in July of 2020, our collaborators published this work and packaged it in a Python code called ClinTrojAn, which is clinical trajectory analysis. And here they demonstrate the initially they developed this work more for biological applications like single cell seq and stuff like that. But then they took this concept and applied it to clinical applications. And here we're looking at, they demonstrated on patients with myocardial infarction. And this is an example of one of their trees that they've developed, where each of the tree terminal points is actually a complication. So what they found is they identified a point in space where you have patients that have negligible complications, and then you can identify trajectories or paths to specific complications such as myocardial rupture, pulmonary edema, congestive heart failure, and you can look at what are the dominant features within this multidimensional data set that sort of contribute to this path. So now to overview of the data, again, we use the COPDGene cohort baseline, which is roughly about 10,000. After some quality control and quality assurance, we ended up with about 9,000 cases. This is a distribution of the data based on gold definitions. So anyone below the ratio of FEV1 over FVC is considered to have COPD. Increasing severity is from right to left. So this is gold 1, mild COPD, and severe. You can clearly see in this data set that a huge segment of the population are those patients at risk. So in a way, that's kind of exciting. That's nice. We did look at 29 features, and this is through consultation with our clinical collaborators. 4 of which are demographic, 7 are spirometric, so that means pulmonary function, 6 are smoking related. We actually included 11 CT-based readouts. Only 4 of them are the ones that we developed that are PRM, and 1 disease classifier, essentially those patients that have bronchitis or not. The first step we needed to do was to determine the optimal number of principal components that the algorithm will use when building the tree. We looked at 3 different techniques. And again, just through heavy discussions with our collaborator, there's a little bit of hand waving here. But for each of these techniques and holding the number of nodes constants, how many points there are in your tree, you can see how the tree changes depending on the number of principal components. So it's very important to try your best to optimize, but to be consistent. So we decided to use the elbow rule, which gave us about 4 principal components. Next, we needed to optimize the complexity of our model. So how many nodes do we want to use in our model? So this is the 100 nodes, 4 principal components. A lot of this work was done by Eugene. He's another collaborator helping us with this project. And what we find, this is the fractional variance explained, and there's a complexity. The rule of thumb is to try to find local minimas that explain as much of the complexity as possible while also explaining variance, or trying to reduce the complexity while explaining as much of the variance as possible. So we settled on this point here, which is roughly around 61 nodes. So here's our final tree. I know you guys were all excited. When's he going to get to the final tree? So this is it. This is our optimal tree. We have 4 principal components and 61 nodes. And right now, this tree is color-coded based on gold status. So green is essentially at-risk smokers. Gold 1 through 4, you can clearly see yellow is gold 2. We could see a worsening, a trajectory right away is clearly evident based on gold. And this map is also includes these gray circles, which the larger the radius, the older the population.
Yeah, because that was my question about the time component. So this is, you're getting into that now.
Right. So this patient population, this COPDGene, the median age is around 50. So all of these patients are roughly around there. So the algorithm is actually finding, so I'm going to jump ahead because I'm actually not going to talk about it in my talk, but I'll talk about it here. What we see, what's really interesting, there's clearly a path going here, and I'll discuss it later. But what is very interesting is there tends to be, at 58, there tends to be this, all of a sudden, this pocket of gold 2 subjects. And in fact, it wasn't until my postdoc included this age range did we see that this is actually, we believe this is just due to an age effect. So as you age, the amount of residual volume within your lung decreases over time. So that's what we believe what we're seeing right here. So the algorithm is picking up this age effect that is completely separate from the disease effect.
Okay, what are the numbers?
So the numbers are, they're branching points. No, they're nodes, right?
Yeah, that just labels identified.
Thank you. So just like in the work Alexander did for the myocardial infarction, we have to determine a node. Essentially, where's our population that appears the healthiest? And we decided on node 40, or that segment. So now from that point, we can look at multiple trajectories, and we can see how our metrics change as we move through this pseudo time. Again, it's important to note this is cross-sectional data. This is like a clustering analysis on cross-sectional data, but it does give us an indication of how things may change potentially as the disease progresses. So what we're going to look at, or what I'll discuss, is, to me, is the most obvious. We're going to move from 40 to 63. So we're going to get to this endpoint, which comprises almost completely of gold 4 subjects. And we'll look at 40 to 59. And the reason why 40 and 59 is that this branch, 59, looks very similar to this branch here. So we're really interested to know what are the differences between these 2 branches, even though their gold status looks very similar. So visually, if we go back to our original PRM technique, this is what the percent of emphysema. So black is 0 and white is 100%. We could see there's maybe some increase in what is perceived as emphysema, but clearly all of the action follows a very specific trajectory. It's, again, emphysema is a one-way path. The algorithm seems to suggest the same as well. But if we look at FSAD, that doesn't seem to be the case. There tends to be a lot of branches that show this small airways disease component of COPD. There's the one we discussed just now, this age effect. But really, again, it's pretty much following the same path as emphysema, but it is important to note there seems to be other trajectories that are worth investigating, and we just haven't gotten there yet. So here is the main trajectory we were interested in, 40 to 63. So this is going from gold 0 to gold 4, and we can follow the x-axis is referred to as pseudo time. 0 is your root node, And these are essentially just nodes moving down this path. So as we're moving down, we see emphysema increases as we're moving along this trajectory, and small airways increases and sort of plateaus. These three bottom are sort of controls. They're what we would expect. FRC is sort of the volume or trapped air in the lung, the volume of the lung at expiration. It should go up as a function, as a worsening severity of disease. The ratio drops. And this is another spirometric measure. This drops as well, just like, so healthy subjects have very good normal functioning lungs. Someone that's diseased, it's very poor. But what is interesting, and it's not something we necessarily see, is, and I tried looking for a data set, but I only found one that showed this, but typically small airways disease increasing is almost linear with increasing gold status. If we follow this specific trajectory, the disease is almost dominated entirely by emphysema. Like there's no more increase in small airways disease that we can see as we move towards this trajectory. And it's almost entirely emphysema, which was actually quite interesting and not necessarily expected.
Goes along with that other analysis at the beginning.
Yeah. So the other target or point of interest is this branching point. And as I mentioned before, as we're moving along this trajectory, you sort of have two directions to take, but they look very similar in terms of gold status. And here they're segmented 4 and 5. These are the actual segments are labeled. So we were interested in determining what are the difference. Pulmonary function is about the same. So the box, just keep your eyes on the box because the labels have decimals, so it's a little harder to read. We do see that there's a slight difference in the ratio, which is FEV1 over forced vital capacity. So those moving in segment 5 tend to have lower values than those. But as far as gold status is concerned, they're both gold 2 in this case. Forced vital capacity was found to be slightly elevated in 5 over 4, and emphysema was especially beginning to rise. So as we're moving down segment 5, we see an increase in the amount of emphysema in the lungs in those patients. But what was really interesting is when we looked at our airway measurements. So this is wall area %, which is a measure of bronchitis, and PRM3 or FSAD is a measure of bronchiolitis, and we can see they're inverted from one another. So in fact, what we're thinking that we're seeing here is actually, if we follow 3 along 4 through this orange arrow, we're actually looking at more larger airway dysfunction. So more bronchitis related COPD, whereas if we move down blue, this is more bronchiolitis, which is the precursor to small airways disease. And that's what this initial investigation is suggesting. So in conclusion, we have demonstrated the potential of clinical trajectory analysis, or ClinTrajAn, to provide a map of COPD progression trajectories using a single cross-sectional data set. So this was not, it was quite large data set, but it was a cross-sectional data set. We anticipated some of the results we sort of expected, that FSAD would precede emphysema. And that is exactly what this unsupervised method showed, or semi-supervised. We did identify multiple branches that were small airways disease dominant, which requires a little bit more investigation on our part. We think one of those may be age related, but some of them we're not entirely sure of why they're there. And then finally, we seem to find a branching point where large airways disease and smaller airways disease sort of diverge from one another and have unique phenotypes and are clustered separately, which also bears further investigation. So ongoing, because this is COPDGene, and 5,000 cases have 5-year follow-up, and we also have the 10-year follow-up, we're really interested in applying the 5-year data set to our current optimized model to see where do they fit in the grand scheme of things, and how did they change over time? Did they follow the trajectory, or did they end up somewhere else? We're also interested in looking at additional classifiers such as comorbidities, bronchitis, ILDs, lung cancer, survival. All this data is available through the COPDGene, and it's just a matter of tapping into it, as well as exacerbations. And then finally, unlike other diseases such as collaborations, deployment-related small airways disease, bronchiolitis obliterans, and lung transplant, they don't have the numbers, the data repository that has been invested in COPD. Can we use these models to infer disease progression in these other disease subsets? Even though they may not be perfectly matched, but they may provide some insights into disease progression, especially for deployment-related lung disease, which is essentially a COPD. So I mentioned a lot of the collaborators in Europe, but the main person who really did a lot of the work and also jumped in for one of my questions is Alexander Bell. He's sort of spearheading this whole initiative, which is heavily appreciated. But I'd like to acknowledge everyone in my lab and my collaborators at Michigan Medicine, as well as Chuck Hatton in Bio, and I already mentioned the others, and obviously the NIH for supporting this work. All right, thank you.
How does fibrosis play into this? I mean,
so fibrosis typically isn't associated with COPD. It's a fibrotic disease. That's why that catch-all parenchymal disease got incorporated in, because we decided to use PRM for other disease applications, such as GVHD in lung transplant patients. You can develop a obstructive disease, bronchiolitis obliterans, but you can also develop a restrictive disease. And we wanted something that could account for both simultaneously. But in the case of COPD, we wouldn't expect that.
I wonder if there's an opportunity to look at what happens when some of these guys along the trajectory get COVID. You know, I mean, I'm serious.
I'm laughing, but it's only because, yes, I completely agree. You know, I'm in talks with some of my collaborators in radiology to try to apply some of these techniques, especially the image-based techniques for COVID cases. But again, you know, one of the big concerns are some of the discussions we just have internally about applying this technique is, you know, we have to be wary because you know, it's like a convolutional network. You're sort of training it on a very specific subset. You know, this is in this case, you're not training anything. It's built off COPD cases. So applying that to IPF patients is obviously not going to hold, right, because so, those cases don't apply.
People on the trajectory. I mean, there's some
right.
like me that are on the trajectory that you might be able to recruit a subset to like, look, I mean.
No, yes. So for me, the ideal data set is, so I collaborate with pulmonologists at the VA, and they're applying some of our imaging readouts for deployment related. So service men and women overseas exposed to burn pits and, you know, hazards out there. You know, they come back and their lungs are not what they used to be. And what we're finding out is, you know, it's a form of chronic bronchiolitis, which is COPD. You know, a data set like this and a model like this could be very advantageous for that patient cohort to get a bit, you know, we just simply don't have the numbers like what we have here, but we may be able to use this data to get a better understanding of what's going on with that very special, specific cohort of patients, population. So, yeah. So I think I got a chat.
So, you know, is there a point of no return? I mean, you know, at some point, you know, it's either, it sounds like you quit smoking or else. I mean, you know, is there like therapeutic, is there a point along the way where there could be an intervention or?
So what we think, and this is a little bit outside my area of expertise, but the idea, emphysema, no. So I heard, actually it was Milan made the point that emphysema is like cancer. It's never good. Once it's identified, it just gets worse. Doesn't matter how you change your habits. It just seems to get worse. It progresses over time. It's down that path. It stays down that path. My understanding of how that's treated is surgical resection. You know, you just have to go in and remove it so that the lungs can actually breathe. So what ends up happening, they inflate and you can't even breathe anymore because you don't have enough space for your lungs to contract. But in terms of a lot of this is, you know, in terms of this small airways disease component, you would think it should be manageable, but like a lot of disease, it's all about early diagnosis. So, you know, a lot of these patients aren't seen. And then when they are seen, things have already gone pretty bad, you know, and that's ultimately the problem is, you know, so we're looking in, you know, I know there's initiatives to sort of, if you're going to do lung cancer screening, can you also include, you know, assessment of an individual's lungs in terms of COPD, because there's a lot of, they have very similar etiologies. They're all kind of smoking driven diseases. So can you tackle two birds with one stone? So I know there's people going in that direction. It's a little bit outside, so don't quote me on it.
So in your trajectory, are there any people
Oh, sorry.
measured multiple times?
Yes. So out of the 10,000, half of them have a 5-year follow-up, and that's what we're working on right now.
Okay, I was going to say, it would be interesting to see where the same patient lies on the trajectory of time.
Right. Yeah. So the goal is, or the plan, and this is what Alex has kind of lined out, is, you know, you can identify an individual and get their distance from the node and then have their follow-up and then take the differences of the two to get like their trajectory. So yeah, we're working on that. So right now that we have to make some modifications to the code to, once we have the model, you know, can we feed new data in so that that data is just presented and doesn't change what the tree looks like. So that's what we're working on right now. So yeah.
I'm just curious, can you talk in a little more detail about what the features actually are that you're using?
Sure. Yeah. So yeah, I think I actually have a, oh man, how do I do this? I do have a table. I'm going to end the show and I'm just going to go straight to the table. There you go. So yeah, so we're looking at age, BMI, pack years, you know, when did you start smoking? How old were you? So FEV1, I'm kind of going down the first column. Sorry, I have a tendency. So yeah, so 1, 2, 3, 4, 5, 6, 7. Those are all spirometric. BDR means before bronchodilation. So one of these patients, to confirm that they're COPD, is you shouldn't get reversal of obstructive, you know, difficulty breathing upon a bronchodilation, because that would be more asthmatic. SGRQ is St. George respiratory score. Distance walk, that's exercise capacity. TLC and FRC are lung volumes. 1 second, sorry. So these are mean densities of lung at inspiration and expiration. Wall area percent, so that's we segment out the airway wall and we measure the wall diameters and the wall thickness. PI10 is inner perimeter at 10 millimeters. That's also an airway wall measurement. And then PRM, this is normal lung, small airways, emphysema, sorry, and parenchymal disease. And this is sort of non-specific. We include it because there are members of COPDGene that are very interested in it. So we included gender, are you smoking, MMRC, there's another DIPSIA score, so breathlessness score, and then exacerbation, which is more of an outcome, race, and chronic bronchitis. So those are all the features that were included.
How do you handle the ones that are not numbers?
What?
Numbers, race, and gender.
So the algorithm does tackle classifiers. So we just now started, because we're using the tree and we're sort of inferring trajectories. But obviously, something like race is not a trajectory. At the end of the day, the tree is in fact a cluster-based approach. I mean, it's grouping like, so these are one of the things that we just recently started playing around with as we're optimizing the tree. We presented the tree, color-coded the tree based on gender, and we found gender differences on the individual branches and the same as race. So it is included in the model, but it's, you know, you can't, right? Someone doesn't change her race halfway down the road. So yeah. So this is a fraction of the total number of features provided by COPDGene. We didn't even put any of the genetic information in there at all. It's all ripe for the picking, I guess you could say.
So we're DNA specimens taken from the patient.
Blood samples. I believe they're just blood samples. I'm not entirely certain. Oh, okay. Yeah. So I'm not entirely. I'm more on the CT side of things.
Yeah, we're more on the other side.
Yes, I know.
Might be a good conversation.
Yes. Yeah. So yeah, how COPDGene works. The main hub, the repository for all CT data is National Jewish, and I can't remember who's, but the genetic information is collected by, and the repository is somewhere else in the U.S. So I'm always in contact with National Jewish, but almost never with the other sites. So, but it is interesting. A lot of the work that Alexander and Andre and Eugene that they did, some of the initial work was all the biology and bioinformatics and stuff like that. So before they published this clinical application paper, they initially had a paper in, I believe it's Nature Communications, that's all single cell seq and how it can be used to identify unique cell phenotypes and stuff like that. So it's nice to see how, especially in the computational side of things, how certain applications and certain computational software applications, how they have very broad they don't necessarily have to be resigned within a certain area of science, but have broader applications.
They're mathematicians.
Excuse me?
They're mathematicians.
Yes, I apologize. They're all mathematicians, so they don't pigeonhole themselves into any one space. They're so yeah. Okay, cool. So just.
All right, thanks a ton.
Thank you.